And welcome back to Basic Pathology. And we are talking about neoplasia, we're talking about cancer. And in the previous videos, we covered quite a lot, so we're just going to continue on, then we'll get into some other things. So just as a review here, transformation and progression, what are we talking about? What things are involved? Of course, if you remember from the lecture, it's self-sufficiency in growth signals, insensitivity to growth inhibiting signals. Evasion of apoptosis, defects in DNA repair, the spell checker. Limitless replicative potential. We're talking here about telomerase. And so we have angiogenesis, invasive ability, metastatic ability. And so here's the question here. We know that most neoplasms can metastasize. What is the exception? What is the type of neoplasm that does not metastasize? That is basal cell carcinoma. Okay, so let's move on. Okay, so we've talked about some of the things that can go wrong. So let's just review some of the things that can go wrong that we should have been doing in the reading. We should know from the reading. So let's just have a look at them. So we have a normal cell, but we can have acquired DNA damaging agents, for example, chemicals, radiation, viruses, and the normal cell can be damaged. It can come back and then it can become normal again, but if not, then it will continue on. For example, when there's a failure of DNA repair. We can have inherited mutations in, for example, genes affecting DNA repair, genes affecting cell growth and apoptosis. There could be mutations in the genome of somatic cells, which could lead to activation of growth-promoting oncogenes, inactivation of tumor suppressor genes, alterations in genes that regulate apoptosis, and we could have unregulated cell proliferation, and we could have decreased apoptosis, all leading to clonal expansion. So then we might have angiogenesis, escape from immunity, additional mutations, tumor progression, malignant neoplasm, invasion, and metastasis. So there we have all the things we've been talking about, just summarized. So let's move on and talk about the molecular basis of cancer. What are the four classes of normal regulatory genes? We have proto-oncogenes, tumor suppressor genes, genes that regulate programmed cell death, and genes involved in DNA repair. We're going to talk about these again. Proto-oncogenes, tumor suppressor genes, genes that regulate programmed cell death, and genes involved in DNA repair. So let's talk about some of these. Remember that carcinogenesis is a multi-step process and no single oncogene causes cancer. So know that it involves numerous categories of genes. So what are the gatekeepers in this process? These are the oncogenes and tumor suppressor genes. What are the caretakers? Who are the caretakers? DNA repair genes. We have tumor progression, angiogenesis, heterogeneity from the original single cell. We need to talk about all of these, know all of them again. What are the gatekeepers we need to know about? Oncogenes, tumor suppressor genes, the caretakers, DNA repair genes. So we're going to see problems in all of these areas. So let's talk about oncogenes, changes in DNA. We're talking about mutations of normal genes, proto-oncogenes. So what does this involve? Growth factors, growth factor receptors, signal transduction proteins, RAS, nuclear regulatory proteins, cell cycle regulators. Oncogenes code for oncoproteins. This is a very important part we have to understand and remember from the lecture. Okay, so what I'll do here is we're going to talk about the associated human tumors. Then we will try to guess the mode of activation and the proto-oncogene and the category. So let's talk first about colon, lung, and pancreatic tumors. What is the mode of activation with this? These are result from point mutations. What are point mutations? These are substitutions, if you remember. What is the proto-oncogene in this situation? This is KRAS. And this is a GTP binding signal transduction protein. How about bladder and kidney tumors? This is also a point mutation. In this case, the proto-oncogene is HRAS. Melanomas, hematologic malignancies. What is this? This is also a point mutation. In this case, it's NRAS. So let's go back over those. How about KRAS, K-RAS? We said it was a point mutation, a substitution problem, resulting in what type of tumors? KRAS resulting in colon, lung, and pancreatic tumors. How about HRAS? Again, a point mutation resulting in bladder and kidney tumors. How about NRAS? Another point mutation resulting in melanomas, hematologic malignancies. Okay, how about chronic myeloid leukemias? What is the mode of activation here? This is a translocation. And the proto-oncogene is ABL. And the category is a non-receptor tyrosine kinase. And the same would be true for acute lymphoblastic leukemia. How about for melanomas only? This is, again, a point mutation. So a lot of these are point mutations resulting from substitution problems. In this case, melanoma is resulting from this proto-oncogene is BRAF. And this is a RAS signal transduction category. How about the hepatoblastomas or hepatocellular carcinoma? This is again a point mutation. And the oncogene here is B-catenin. And this is WNT signal transduction. Are you going to be remembering all of these for the test? I don't know. There's a good chance. That's why we're talking about them. So what are we taking out of this? Well, we certainly seem to have a lot of point mutations, substitution problems. So let's move on. Let's talk more about carcinogenesis. Remember that it involves initiation, which is a mutation, and promotion, which is proliferation. So we have mutation, and then we have proliferation. So how does this system work? How do we have unregulated cellular proliferation? I need some sleep. Remember that the activation of oncogenes is activation of oncogenes by overexpression, mutation, chromosomal translocation results in unregulated cellular proliferation. So we have overexpression, they're too active, there is a mutation, and there are chromosomal translocations. So let's think about breast cancer. So in this case we're talking about epidermal growth factor receptor. We're talking about ERB-B2. There's an amplification here resulting in breast cancer. How about neuroblastomas? There is an amplification here. We're talking about N-MYC, which is nuclear regulatory protein. Okay, and next, how about glioblastoma multiforme, GBM? We're talking about CDK4 cyclin-dependent kinase, again an amplification. So I hope you memorize all of those. So here's the funny thing about this. When you take the time to read out a textbook or read out certain difficult parts of a book or from a lecture, you actually end up learning them. That may be just the case for me, but just once or twice reading through using my voice actually results in me learning all of this. I'm able to recall them. Anyway, that's why we're doing this. If it doesn't work for you, I'm sorry, but it works for me. So let's talk about chromosome changes in cancer, translocations, and Inversions. What do these occur mostly in, these translocations or inversions? They occur mostly in lymphomas and leukemias. They occur in many of the non-hematologic malignancies, so remember that.